T cells talk about the lymphatic system. Now this slide is showing you both B cells and T cells, which again is a good reminder after the weekend. I talked about the innate non-specific defenses, about the surface barriers, the membrane barriers, and the second line of defense, the phagocytes, but then the adaptive defenses, the sensitized lymphocytes, are shown here. Talked about the B cells. They can actually bind the free antigens directly. Remember that. B cells are stimulated by direct binding of, as the figure says, the three AGs, antigens. AG is short for antigens. 2 membrane-bound antibodies. ABs as antibodies, and that acts as their receptor. And we talked about the whole thing. They're proliferated. That clone is proliferated to fight, and it'll produce plasma cells, which manufacture the antibodies, and some hang around in your bloodstream to be memory B cells, which can mount a faster, stronger response. Now, the important thing about this figure is, well, how do the other T cells come into play? We're going to see that today. Here, they're activated differently. What you see here is, this is some kind of phagocyte. In this case, it's a dendritic cell. And this cell has to present a piece of the pathogen to the T cell to become activated. Here, it can bind an antigen directly, the B cells can. But the T cells need help. They need a cell called an antigen presenting cell to literally present an antigen to activate and stimulate. Okay, so I think that's the main thing to take away from this figure, is the difference of how they're activated. T cells are stimulated by an antigen presenting cell which, as the descriptor implies, stimulated by an antigen presenting cell, which presents, quote unquote, a piece of antigen to the T cell. And if you remember, there's different subpopulations of T cells. There's CD4, CD8, helper killer, or helper cytotoxic T cell. They'll kind of proliferate in. Another important thing to take away from this figure is the crosstalk. There's crosstalk between the memory, the T helper cells and B cells. They help activate it. Okay. TH, those are the T helper cells, can help activate B cells. So they don't operate independent of each other, they are dependent on each other. You depend on the T helper cells to kickstart the B cells and everything else for that matter. Okay, we'll go through the details of that. Now, so just to focus on T cells now, let's be reminded of the two main subpopulations I want you to know, CD4, CD8, for T cell mediated immunity. So what this figure shows you is some things we've already discussed, like for example, all formed elements, white blood cells, red blood cells, or platelets, including the T cells, are formed in the red model. However, what we'll see today is that the thymus is also important for the maturation process of T cells. Okay. So we'll need to look at that. Okay. The thymus is important for T cell maturation. Okay, the other thing that's important from this figure is showing you the two subpopulations, CD4 and then CD8, also known as T helper and cytotoxic T cell. So it looks like on the left there, you have some cells that become the T helper cells, or CD4. T helper, or also called CD4 cells. On the other side, looks like you have cytotoxic T cells, TC, or the CD8 cells, and again, they're activated by the APC. Okay, APC is the antigen-presenting cell, so both needed. Okay, you need the APC. You need the APC, and also this is a function. Antigen-presenting cell is a function, and only certain cell types have this function, and they include macrophages, cells called dendritic cells, B cells can have this function. So those are cell types that have this APC function. And when they present that antigen, they do so in a way, there's different subclasses. There's proteins on the APC cell that can interact with either CD4 or CD8, and they're called the Mhc proteins, and there's different classes. Mhc class 1 is restricted to interact with CD8. Is Mhc class 1 proteins are restricted to interact with the CD8 cells, whereas on the other side, the Mhc class 2 proteins are restricted to interact with the other ones, the CD4s. Mhc class 2 proteins restricted to interact with CD4 cells. They're like proteins that help present the antigen, and they're very important to the activation of the CD4s and the CD8s. MHC is an acronym for Major Histocompatibility Complex. I'll write it up here. Major histocompatibility complex. It's a mouthful, which is why we typically use MHC. Sometimes also called human leukocyte antigen protein. You may see that, but I tend to use MHC. So they're important for immunity. So I have a slide here to remind us about a term called Mhc restriction. Whenever you see that term, Mhc restriction, it's a concept really. CD4 and CD8 cells, they bind to different classes of MHC. We already went through it, the class 1 and the class 2. The class 1 MHC, CD8 cells, they can become the cytotoxic C cells, and while they're activated by antigen fragments linked to class 1 MHC. The class 2s, CD4 cells become helper T cells. They bind to antigens linked to class 2 MHCs. So Mhc restriction is simply the class 2s are restricted to interact with the CD4s. The class 1s are restricted to interact with CD8. In other words, does MHC1 ever interact with CD4? No. That's why I kind of made two sides of the board here. So that's the idea. It's pretty easy. Now, to appreciate MHC, major histocompatibility, histo, does that sound familiar? Yeah, that's a good guess. Was there a chapter called Histology?
Yes.
And what was that chapter on?
About
Tissues. Histo is a word root for tissues. So what are these histocompatibility? What does it mean to be histocompatible? It helps to know as a student the history behind how they discovered these proteins. I can't remember what day it was. I think it was the 70s. They would do these experiments on inbred mice. Now, if you don't know, the mouse model is very common in research. And the mouse model, what they do is they take mice, a litter of mice, and they inbreed them. Siblings mate. And when you do that, you have a pure genetic strain. Okay, because the genes are only from the two parents. If all the siblings are mating generation after generation after generation. You do this with mice, right? Not humans, we don't do it. But mice, you do it. And it's a great model. Because when you have an inbred strain, you have a genetically pure strain. And if you have two strains that are genetically pure, what they would do is they would take a tissue graft from one strain and they would put that graft and try to graft the skin onto another strain of mice. Now, what do you think happened? Was that tissue graft accepted or rejected when you go strain to strain? Rejected, right? And so the question was, what are the class of proteins responsible for the tissue graft rejection? Because if you put a tissue graft within the same strain, it's accepted. Okay, so they're trying to figure it out. Well, long story short, what they found was there's a complex of genes responsible for the tissue graft rejection, and they call them histocompatibility complex. And then further research showed the genes with the most potent effect for the tissue graft rejection were called major histocompatibility complex. So these proteins are responsible for tissue graft rejection in those studies. So the name stuck, right? So it makes sense, right? We study it in immunity because these proteins help reject anything that's non-self in your body, especially a tissue graft. So that's where the term comes from. And we use it here because we're teaching immunity. And so let's take a look at class 1 first. Class 1 MHC. And so I kind of came up with this list when I'm trying to make it make sense to my brain. So, these are the key questions that I think can help you understand it as you study it, to try to understand how they work. Questions you can ask yourself. Okay, well, the first thing on the top there is where we established the restriction. The class 1s interact with the CD8s. You can just remember that. Then, which cells present the class 1 MHC? Okay, that's good to know. All cells, except cells without a nucleus, because you can't make proteins without a nucleus. So like RBCs don't have them, but everything else does. Which cells can present class 1 MHC? It's like all cells, not RBCs. They don't have a nuke. I read that, I'm thinking this is like your signal to your immune system. This is me. This is self. You don't attack self, okay? It's like if you're on the battlefield, and you have an American flag on your tank or whatever, on your uniform. Everyone on your side, they don't fire at you. It's you. You're on the same side, right? It's kind of like that. It's like your body has this class of MHC, and your immune system comes around, it's looking for trouble. Don't attack me, I'm you. And if you attack yourself as an autoimmune response, then that's not good. All right, so what do they display? They display a piece of yourself. Call it an endogenous antigen. So those MHC1s display MHC1. They carry a little piece of endogenous antigen. I'll call it endogenous AG, right? So what kind of antigen is it? It can either be self or non-self. So the endogenous AC can either be self or non-self. If you're well, you don't have any infection, it's always going to be self, right? Let's say you get sick. And some of your cells will contain virus or whatever, and then it may display a piece of that endogenous non-self, and you want to respond to that if you're sick, right? So non-self, say you got an infection, you're sick, infected. Okay, so that's kind of the story with the CD8s, class 1. So here's a picture of that to kind of bring those points to life. So we're looking at a cell presenting these endogenous antigens. And so we have a cell that's presenting class 1. So what kind of cell is it? Any cell. So just not RBCs. This presents the class 1s. And what it does is it just takes a piece of itself from within. Endogenous means from within itself. Takes a piece, puts it on that groove, and displays it. So white blood cells come along, and it kind of fits in a way and say, oh, self, I don't activate, I move on, Self, okay, I move on. This cell's okay. Right? That's kind of what it is. Okay, any questions on this information here? We're going to do the other ones. Class 2. Well, same kind of questions. Which cells can present the class 2s? These are only the APCs. Only APCs present class 2 MHC. So, let's see here. Okay, so I'm going to put this list right there. I don't want to confuse you because I have it listed here under this one, and I say all cells. All cells could include an APC. So to be less confusing, let's just hit all cells as a better answer and cross this out. Let's remember why I put it there in the first place. I was teaching from this figure. See how they put APC on both sides? But it could be an APC, but it's all cells, all right? So that's why I put it there. So for the class 2s, to be more clear, only APCs present class 2, okay? So, and let's be clear what the APCs are, those three kinds of cells. It could be either the dendritic cells, the macrophage, or the B cells. Dendritic cells, macrophages, B cells. These are cells that have this APC function for the class 2. And what do they display? Well, it's always going to be an exogenous antigen. And so by default, that exogenous antigen that came from outside the cell is always going to be non-self. An infection you want to get rid of, always. So, MHC class 2 display an exogenous AG, it's always non-self. And so they give you a nice picture of that in the textbook, where they show you one of these three cells, which all have a phagocyte function. What do phagocytes do? They're always trying to find things that are scavenging for garbage, and there's kind of like an antigen that's a pathogen. So you engulf it, phagocytosis. It's phagocytized. And what you do is you break it up into parts, and you put a piece of it into the groove of the class 2, and you display it. Okay? So a CD4 comes along and activates it. That's the goal. You got to find this cell. And when you find it, it has to bind in such a way to recognize, to be specific for that viral, that bacterial AG that's presented there. So let's talk about that. Let's start with the T cell activation, particularly of the CD4s, or the helper T cells. Yeah?
So when you talk about class 1 MHC, so that displays endogenous antigen, so that's only for self, or it's for non-self? Because you wrote in the bottom that says I'm infected, so it's with the self.
Okay, yeah. So let's go back to that figure. So I think this is kind of what you're getting at. I said they present an endogenous antigen. If it's a piece of itself, the piece of the cell that's from the cell, it's considered self. But let's say this was infected by a virus. And imagine this is a piece of virus that's now displayed. That would be non-self. You got it?
The infection will be non-self, but the cell is self.
That's correct. If you're not sick, it's self. If it's virus infected, it's non-self. Okay. About this case, always non-self, because it's always something that's phagocytized by one of those cells. All right, so now I'm talking about the activation of a CD4 by such a cell. Okay, that's why we do this. Now here's an outline of how the CD4 recognizes it. All these have to happen. And this outline is better understood by looking at a picture like this one. Okay, here's an APC, right, presenting the class 2 MHC. It is interacting with a T cell, a CD4 helper T cell. And so all the things you should recognize here, it helps to kind of draw it out. So I'll erase this now. APC cell. It's presenting class 2 MHC. It's class 2 MHC. And the other cell is a helper T cell. CD4 T helper cell, and it has a receptor to match it. It's TCR, T cell receptor. Very easy. Now, let's remember, the APC is presenting a viral AG. Okay? So that viral AG is specific for the TCR, right? This is adaptive immunity, specific immunity. So this reaction happens because this TCR is specific for that antigen, and this is a CD4 cell, and it's restricted to interacting with MHC class 2. And to help stabilize this interaction is a protein called, I'm mentioning it, using different color. CD4. So this is the actual CD4. Okay. And so this reaction, MHC class 2, TCR, specific for the viral AG, boom, stabilized by CD4. That sets off like a signal, called like signal 1. But that's not enough to activate it. You really need a co-activation to happen to ensure safe activation. So not just one signal, the second signal. The co-activation, there's these other ligands. This one's called B7 on the APC. And then the CD4 presents something to interact with it called CD28. That's like a co-stimulation, and it sets off a second signal that will ensure proper activation. And both of those signals combine for like a complete activation. Like for example, an activated cell will start to present other surface proteins. Like for example, this one here I'll draw, CD154. CD154, which otherwise it wouldn't be there. Evidence that it's activated. And lastly, as you can see, there's other cytokines that help these signals jumpstart, and that is interleukin 1. Just call it IL1. That's really what you see here. And if you go back to the previous slide and compare the picture to the outline, it's everything you need to know. The TCR recognizes the MHC class 2, but also is specific for the viral antigen, right? That particular bug. You have a clone against that particular bug. And like I say, CD4 stabilizes that for signal 1. Then for number 3, the B7 CD28 is referred to as the co-stimulation, right? And interleukin helps. Interleukin 1 helps. And the name of the cytokine is easy to remember. It's literally a molecule secreted between, leukocytes. Okay, so now all you've done is you've recognized and you've activated. And what are the things that happen as a result of that? And I have another picture of this. This kind of illustration here. I like this one because it actually shows both signals being jump-started there at both signals. An activated cell will now have other proteins like CD154, and an activated cell will now secrete other interleukins like IL-2 to help kickstart other lymphocytes to become activated. And to emphasize the importance of this, yeah, question?
Yes. What is TC4? TC4.
Anyone remember?
No.
TCR?
receptor.
T cell receptor. Yeah, you might want to write that in your notes. So any other abbreviations up there? Any clarification? What's APC? Antigen presenting cell. I don't know what the B stands for. Don't ask me about B for B7. CD28. CD always stands for cell differentiation. IL is interleukin. Yeah. Okay. Yeah, a lot of abbreviations in science. Yeah, keep track of them. Now, without this co-stimulation, something called anergy occurs. Basically, nothing happens. Okay. T cells, they become tolerant to that antigen, meaning they just see it, but they don't do anything. They don't divide, they don't proliferate, they don't secrete cytokines. Basically, no immune response, nothing. Okay, so that co-stimulation, that second signal, is very important. Okay. Like if this were a nuclear missile, you want to arm the nuclear missile, you need two keys to activate it. Both keys need to be turned. Okay, otherwise, no action, no reaction to the antigen, and you can't mount an immune response. So, all right, let's say you properly activate it. Actually, how did this CD4 cell help? Help is a good term. Here we see it doing a couple of things. Here's the helper cell. Here how it's activating a couple of other cells. Okay, first, the T helper cells, it's binding to this APC, this dendritic cell. And it stimulates the dendritic cell to express the co-stimulatory molecules, right? You know, like the CD28B7 stuff. So how do helper cells help? T helper cells help. They help APCs. They may help to express the co-stimulatory cells. Help APCs to express co-stimulatory molecules. Like the B cell, okay, something like that. Secondly, the dendritic cell is now activated, and the CD8 cell, with the help of IL-2 from the T helper cell, can interact with it. So the T helper cell help the CD8 cells with the help of IL-2. You can activate the CD8s. IL-2 can activate CD8 cells. So they're helping both the APCs and the other kind of T cell populations. And like I said before, I'll say it again, they also help activate B cells. Here's IL-4. The T cell releases IL-4 as co-stimulatory signals to complete B cell activation. So they help the B cells activate too. So the T helper cells help B cells. helps to complete B cell activation. So again, the CD4s are the linchpin. You get everything going, you're good. Now the cytotoxic T cells, what can they do once they're activated? They can administer what's called this hand-to-hand combat, okay? They target the virus-infected cells, and they apply what's called a lethal hit by releasing perforins and granzymes by exocytosis on the virus-infected cell. So here's a picture of that target cell, which is virus-infected, and there's the activated T cell, the cytotoxic T cell. So you remember these are the CD8s. I'll say when activated, can apply a lethal hit to a virus-infected cell with what are called the perforins and the granzymes. So those are the two key molecules there to take note of. Perforins perforate. Okay, there's the perforin pore, and the granzymes, they kind of get in. Granzymes enter the targets via target cell via the pores. What's inside these proteases degrade cellular contents, stimulating apoptosis, okay, destruction of the cell, a lethal hit. All right, so that actually concludes immunity, and I inserted some practice questions in addition to the study guide to help you study as you get ready, and even some blood typing review slides. I think you should be ready for that. And, remember, the written part of the exam does cover blood typing, so I want you to be ready for that. But the exam also covers lymphatics. So this concludes immunity, but I should do lymphatics next, as that is also on next exam 5. So I'm opening up a new batch of slides. It's a different chapter, but it's related. The lymphatics is basically part of your immune system. It's what the lymphocytes operate in. Lymphocytes are the cells of the lymphatic system. So let's study the whole system. First of all, there's kind of a dual function here. There's the lymphocytes, the lymphatic system, which have immune function, but then there's lymph, which actually is part of the circulatory system. What is lymph? Clear the board here. Lymph is fluid. It's the fluid that's picked up from capillary exchange. So essentially, that fluid, since it's from capillary exchange, is filtered blood plasma interstitium. Now the picture is elephantitis, an infection of the lymph nodes. Well, basically it's a picture of what happens when lymphatics fail. You can't pick up the excess, or if you do, it's all clogged and it can't be returned to the blood vessels, so you swell. And so lymph is generated by the hydrostatic pressure in the capillaries, right? That creates edema. When lymphatics are working normally, the lymphatic vessels have leaky walls which pick up that excess fluid to prevent edema, swelling. Okay? Now all the body's lymph, it drains into these ducts. The largest vessels in lymphatics are called ducts. Okay? Talked about the blood vessels. Lymphatics are part of the circulatory system. They have vessels too. Largest vessels in lymphatics are called, ducts. And there's only two they have to keep track of: the thoracic duct and the right lymphatic duct. So that's not so hard. Right lymphatic duct, thoracic duct. The smallest vessels in lymphatics are called capillaries, not to be confused with the blood capillaries. You have lymphatic capillaries as well. The largest vessels, the smallest vessels. These capillaries are very leaky. They can pick up fluids in the tissues quite easily. So here's a picture of this, what's like a blueprint, a schematic of how it interacts with the blood capillaries. So what you have here is at the capillary exchange, the green is lymphatics, and lymphatics are not green. That's just the color that they choose to use. If the cell of the lymphatic system are white blood cells, what color should they be? White. Okay, but they just use green. I guess it looks pretty. All right. You pick up the excess fluids here, and then, like veins, lymphatics don't have access to the heart to apply pressure to it. They have all these valves to prevent backwards flow of blood. But like veins, you move around and you kind of squish them, compress them. You squeeze it in one way. Except, unlike veins, lymphatics have lymph nodes along the way. We'll look at it. It's basically got a capsule around it, and there's lymph tissue inside there. It's packed out with white blood cells. So if you have an infection in your connective tissues, it's likely that it could be picked up by the lymph. And then you filter it through nodes packed out with lymphocytes. It's a good way to recognize invaders in your body. Okay, so that's why it's also part of the immune system. And look what happens as you kind of squeeze it through lymph node after lymph node. You finally get it back to the blood circulation. So lymphatics are considered, they're part of the circulatory system because they return the lymph to venous blood. Lymph, which is fluid, remember, lymph is fluid, is returned to venous blood. And so I'll put in parentheses, consider this, you're contributing to the blood volume, right? You're part of the circulatory system, part of circulation. For example, I've heard that if a woman receives a mastectomy, a lot of times they remove the lymphatics around here to make sure that they removed it. They're not sure the cancer is spread there. So without lymphatics there, it helps to sleep with your arm elevated. That helps the drainage because you don't have the lymphatics. So it is very important for circulation. Here's a picture of some of the anatomy I want you to know for the system. I already mentioned right lymphatic duct, right there. Here's thoracic duct. Okay, pointing to that green structure right there, draining right there. I would also write down cisterna chyli. That structure is receiving all the lymph from the lower body. And then when it receives it from the lower body, it's returning it, it's continuous with thoracic duct. So you go straight from cisterna chyli, delivers right to thoracic duct. So you can find the thoracic duct. It's located in a body cavity called the mediastinum. All right, also, there's very important groups of palpable lymph nodes. They're all over the place, but I'll have you know the lymph nodes in cervical region, the axillary region, and the inguinal region. So I'll also write that down in terms of like anatomy structures. Lymph nodes. The cervical region, the axillary region, and the groin region, inguinal region. Here's a picture of the mediastinum. You can see cisterna chyli inferiorly, right below the diaphragm there. It receives all the lymph from the lower body, kind of drains up through here, continues with thoracic duct. And thoracic duct will drain right there along with the right lymphatic duct into what's called, a closer picture of it here, the venous angle, where the subclavian vein merges with internal jugular vein to become brachiocephalic vein. When it is right at that angle is where you get the drainage, drain into. So, you got thoracic duct, right lymphatic duct, okay, both drain into what's called the venous angle. It's where IJV merges with the subclavian vein, SCV. So remember your blood vessels. All right, so let's kind of take a step back and let's look at how the lymph capillaries pick up the lymph in the first place at the smallest level, at the lymphatic capillary level. And what you can look at, there's primary and secondary valves in the lymphatic capillaries. Answers the question, how do lymphatic capillaries pick up lymph? how do lymphatic capillaries pick up lymph? I'll phrase it in the form of a question. First, look at primary valves, sometimes called flap-like mini valves. So these are valves that are flap-like. They're mini valves, they're not true valves. So what you see there in the picture is the capillaries, like other capillaries, the blood capillaries, they're overlapping ECs. They're cells that overlap. So it's like you have a cell that overlaps. What you do is like one end of the cell that's overlapping with this one is anchored, okay, by connective tissue. That allows this other one, which is not anchored, to flap open. So this one can kind of, you can flap open. When it flaps open, fluids can enter. That's how you get in through the primary valves. And once you pick up the fluid, how do you move it forward? And I inserted this slide to help teach us that. The top is kind of what I just talked about. It's pretty good. It shows you the overlapping cells. The bottom cell is kind of flapping open, allowing fluid to enter. They call this the expansion phase. You don't have to know that. But anyways, when you compress the structure, it collapses the primary valve, and you push the fluid forward through these internal valves, okay, or secondary valves. So compression of lymphatics compresses, it closes the primary valves. Therefore, pushing lymph forward through the internal or secondary valves. Similar to the venous valves. You're inside, you get pushed forward. You can't go backwards once you're pushed forward because they close like the semilunar valve. So that's how lymphatic essentially moves. Now what I want to do is kind of take a look at all the lymph tissues and lymph organs that you have to know. Lymph tissues are simply lymph tissue just without a capsule. Okay? We're going to look at lymph organs. The only difference is an organ is lymph tissue with a capsule, lymph tissue, no capsule. And they're strategically placed in different parts of the body. They're going to provide a proliferation site for lymphocytes and like a surveillance point, okay? You're looking for the bad guys there. So proliferate and just monitor. All right. So lymphocytes, they kind of temporarily reside in lymph tissues. They can leave to patrol the body, especially if they get activated by antigen. All right, so let me try out the picture there. Study lymph tissue. The main tissue of lymph tissue is reticular tissue. The main fiber of reticular tissue is reticular fibers. They form a sponge-like mesh, and all the red cells are presumably lymphocytes. or phagocytes are listed here. Cells in the immune system that have APC function or are basically B and T limbs. Okay? So if your job is to find infection and fight infection, you need to slow the lymph down. So as lymph flows through you, force it through a mesh. That'll slow it down. If there's a pathogen in there, it's more likely all these cells that are in the mesh can bind it and find it. Okay? So that's why particular tissue is the tissue of lymphatics. Particular tissue is the tissue of lymph tissue. Forms a sponge-like mesh. Particular fibers form a sponge-like mesh. So all the cells there in the mesh. Well, here's a picture of lymph tissue, an example. Let me turn the lights off. Look for the purple. This is in the small intestine, the wall of the small intestine. We haven't had digestion yet, but people know digestion is for the absorption of food. And if small intestine, the food is flowing along the top there, all the digested food gets pulled through the mucosa and through the whole thickness of the intestinal wall. And inside the intestinal wall are these big purple splotches. And those are presumably lymphocytes in the lymph tissue. It's called Malt, mucosa-associated lymphoid tissue. This example in the small intestine. Write that down. Malt is lymph tissue. Mucosa-associated lymphoid tissue. And so as the food stuff is pulled through, if there's any infection in there, it can be found here. And so all the stuff that comes out that goes through are called efferent lymphatics. Remember, afferent goes in, but efferent is coming out. Pay attention to efferent lymphatics. After the stuff comes out, if there's an infection in there, there could be a white blood cell in there that's been activated. That's part of the efferent lymphatics. You have to have a way in, you have to have a way out. So it's good to have it in your gut. I mean, you don't know what you ate. A fly lands on your food. Where's that fly been? You ate it, and it goes through your digestive tract. Hopefully it doesn't make you sick. But you have lymphatics to help fight the infection. All right, capsule present, lymph organs, lymph nodes, spleen, thymus. Okay, so these are examples of lymph organs. Malt is an example of a lymph tissue.
I have a question.
Yeah, go ahead.
So, you're saying that the efferent lymphatics are what's coming out of the intestinal lumen?
Yeah.
Okay.
No, well, no, not the lumen. Let's go back to that. Pretend there's blood vessels here,
Okay.
or lymphatics. Either of that. The stuff that comes out of the lymph goes into more lymphatics or blood vessels. Okay,
And
part of the
that's
circulation.
efferent lymphatic.
Yes, yes. So to study some of the lymph organs of this system, there's a capsule present. I already mentioned lymph nodes, but spleen and thymus are also part of this system. Let's go through those. Here's kind of the structure of a lymph node. So again, lymph organs include lymph nodes, spleen, thymus. This slide is showing you the basic anatomy, the basic structure of a lymph node. Structural organization of a lymph node. So again, you have to have a way in, way out. There's afferent lymphatics, and there's efferent lymphatics. And if you look at the valves, the internal valves favor a one-way flow through this lymph node. So it's a kidney-shaped structure. If you look on this side out here on the left, the valves favor one-way flow in. So this is afferent lymphatics. And I put this picture here to remind me that you have lymph nodes in a row, more than one lymph node in a row. They're in series. So for example, this first lymph node can send lymph to a next lymph node. Lymph nodes are the only structures that can receive efferent lymphatics from a previous lymph node. Okay. Lymph node can't receive efferent lymphatics from a previous lymph node. And that's great. Think about it. You're a pathogen. You're trying to survive in this host that you've infected. But it's like you're putting through this gauntlet of lymph nodes. You're like, there's all these white blood cells. Like, oh wow, I made it through. Oof, oof, I'm still alive. You got to go through another one, and you got to go through another one. Okay, until someone recognizes you and kills you as part of your immune system. So it's good to have this arrangement. All right. So the organization that flowing through one is you got the afferent lymphatics come in on this side. So afferent lymphatics come in. I'm just writing afferent lymphatics enter lymph node. The lymphatics can do one of two things. Lymph can flow one of two ways. Okay, look first at the outer cortex where the afferent lymphatics are coming in. there's all of these lymphoid follicles. Part of the structural organization is you have trabeculae, connective tissue that kind of folds in and creates all these compartments. So the first thing to note is that the trabeculae form lymphoid follicles in the cortex. That's the first thing I'll note. Trabeculae form, I'll call it lymphoid follicles in the cortex. Remember, cortex is always the outer region. It's the outer region where the afferent lymphatics are coming in. And notice what a lymphoid follicle is. It's a germinal center, so you can get the proliferation of WBCs, okay? So you have a lot of white blood cells there. Follicles may contain B or T cells, maybe some dendritic cells. They're proliferating there, they live there. It's kind of their domain. So you have these follicles there. I mean, the lymphatics is coming in, and the lymph can flow one of two ways. It can flow through the follicle, I would say this is desirable. If there's an infection there, you want it to see these cells for an encounter, okay? Or it can kind of escape and flow around it, right? It's flow through, flow around. In either case, you flow through, flow around. You get to this inner part of the lymph node, called the medulla, and there you have medullary cords, medullary sinuses. Now the cords are cords of primarily T cells. Okay? So it's another place where you can have an encounter. So you get the cortex, then go through the medulla, where you have medullary cords, and remember the cords, I'll put T for T cell, that's the primary lymphocyte that's there, another place. Or you can just, again, flow around that and escape that. You can flow through the spaces around the cords called medullary sinus, where there are no lymphs, and you can get out. So you have all these spaces where you can flow through, flow around. When you flow around a follicle like that, the space around it is called trabecular sinus. So I should have mentioned that previously. You can flow around the follicle in, what is it again? Oh God, my brain's full. Yeah, thank you. Trabecular sinus. So you can flow around it the first time, the trabecular sinus, get to the cords, you flow around the cords in the medullary sinus. If you're a pathogen and you make it through, you get out, okay? And then you get out of here as part of efferent lymphatics. But then you might have to flow through another lymph node, because there may be another lymph node that receives the efferent lymphatics from a previous one. So that's a basic idea about lymph nodes. Simply put, this lymph node is filled with reticular tissue, and the stuff is just oozing through very slowly, looking for infections. And you have an organ that actually does the same thing for blood. So the lymph node is to lymph as the spleen is to blood. It's the largest mass of lymphoid tissue in the body. Like I just said, the lymph node is to lymph as spleen is to blood. So the spleen will fight any infection that is bloodborne. But also, because it's filtering blood, it's a good place to kind of like remove like red blood cell parts. I think of it as a red blood cell graveyard. It's where the old red blood cells go to retire and die. And you recycle the parts, like iron. But also, it's packed out with lymph tissue, so it's a great place for an immune response. There's a picture of there's a picture of a spleen. Where is it? There it is. I'm just going to write filters blood. And fight blood-borne pathogens. Location, basically, left of the stomach. Located, it's intraperitoneal. It's not behind the body wall. It's actually easy to find. Located intraperitoneal, left of the stomach. Left side by stomach. The picture on the left shows the full stomach there with the spleen next to it. The picture on the right removes part of the stomach so you can see the splenic artery and splenic vein, which is the blood supply. You should know that. So no. Splenic A and B. Okay. That artery, the delivering blood to the spleen, it's gonna like supply all the blood that you're filtering, trying to find a bloodborne infection. Okay. If you cut the spleen open on fresh dissection, there's whitish tissue that is your, well, they call it white pulp. There's white pulp and red pulp. White pulp referring to the white blood cells that perform immune function. Red pulp is all the RBCs. Okay, it's like some make it through back into circulation and some go there to die. Like I said, it's a red blood cell graveyard. We call it white pulp, red pulp. Now, on a histology slide, the white pulp stains purple. So the white blood cells usually take up the purple stain. So WP, white pulp, RP, red pulp. There is a capsule. Okay. The capsule actually has some function. It's not just a capsule. There's some smooth muscle in there. During a sympathetic response, that capsule can squeeze the spleen. You can squeeze a little bit more blood out of the spleen for a sympathetic response. Another picture of white pulp. Remember when I taught blood vessels and I wanted an example of arterial? One of the slides to look at today is the white pulp surrounding a central artery, or arterial. T-I-T-M, no T-A. Remember that? That arterial is surrounded by a cup of white blood cells. So if you've got a bloodborne infection, it could be, that infection could be found by the white pulp. Here's a better picture showing you the circulation of the spleen. This is kind of what I want to teach from here. So there's all these abbreviations here, which I'll spell out for you. And what's special about the spleen is an example of open circulation. Normally, circulation is closed. Blood flow is normally arteries, capillaries, then what? Veins, and then what? Back to the heart, right? And it's the same thing here, except there's a gap between capillary, and vein. So let's kind of back it up here to splenic artery. SA, blood coming in. The SA is right there, that's where I'm starting. Splenic artery. There's branches of the splenic artery. It branches out and it branches out. It kind of shows you that. Branches, branches, branches. And it branches to a small arteriole. The little a is, call it a central arteriole, central artery. Call it central arteriole. It's a small artery, and it's surrounded by a cup of white blood cells called PALS, P-A-L-S, which is an acronym for, so central arteriole surrounded by PALS. P-A-L-S stands for periarteriolar lymphoid sheath. Periarteriolar lymphoid sheath, PALS, a cup of white blood cells around an artery. We called it white pulp just on the previous slide. Same thing. It's just if you take a slice of it, right there, and you look at it, you see a little artery surrounded by white pulp. You know, that's the white pulp. Periarteriolar lymphoid sheath. Okay, so you got the central arteriole. It gives off these little branches. That's a PA. That little branch coming off the central arteriole is a little artery called penicillary artery. It's like it's even smaller. And what you got there is this little penicillary artery leads right into this end sac called a sheath capillary. So they got the PALS, primarily helper T cells in here. And the penicillary arteries lead to this, that says SC in there, which means sheath capillary. So the penicillary arteries are PA, and the sheath capillaries are SC. And well, what are they sheathed with? The sheath capillaries are different. They're sacs. They have a sac shape. And the sac is lined with like macrophages. An end sac lined with macrophages. The macrophages are illustrated by the yellow sac around the sac-shaped capillary. Okay. I mean, so think about it. You're a pathogen. You got to drive down this street. It's lined with helper T cells. You could have an antigenic encounter there. You make it through that. You go through. It's like a dead end of macrophages. You got to make it through that gauntlet. Okay. So the spleen is very good at fighting bloodborne infections. Now here's the thing. Usually capillaries are continuous with veins, not in the spleen. There's a gap between the sheath capillary and the S, which is a sinusoid, which is venous blood. The blue is venous blood. And so they put RP there. I mean, RP is everywhere. it's good they put it right there. They want you to notice the gap between the capillary and the vein, basically. So I'm going to continue here, from there to here. So there's a gap. It's basically RP, red pulp. All the blood has to filter out of the sheath capillary through the macrophages into the red pulp. Okay? Then you get S is for sinusoid. And the sinusoid is basically venous blood. The little tributaries will merge, and eventually you get to the splenic vein. Then you're on the way out. So that gap is the reason why I said earlier, this is an example of open circulation. All the blood has to flow out of the capillary into the spleen organ. And then some of those red blood cells will make it back into circulation, some will not. If you look at another picture of the red pulp, I kind of like how they show this here. So on this figure, the sinusoid, they use red now, it's called red pulp sinus. So here's, pay attention to this, here's the capillary and the yellow SC. So the yellow is like macrophages. All the blood has to filter out. Okay, so the yellow is a filter. So anywhere there's not a filter, they call it NFA, a non-filtering area. Just pretty much where the SC is not. And all the blood filters out and gets into the blood. However, young, vibrant red blood cells, they're flexible enough so that when they get filtered out here, they can bend and flex and get back in the venous blood. Old, aged red blood cells are more stiff. They're not flexible enough to navigate through the sinus wall, so they just kind of stay here to die. Okay. We recycle the parts. Call it, like I said, I call it a red blood cell graveyard in the spirit of Halloween, right? So that's where the old red blood cells go to die, and that's why you have red pulp in the spleen. So any questions on the circulation? This is the key to understand here. Open circulation. Okay. So again, the open part is right there.
I had a question.
Yeah, go ahead.
You said that some of the young blood RBCs escapes and goes to the non-filtering area, and then it can squeeze itself in the red pulp.
Yeah.
So can that say that if they might be infected but they go back into the cell because they can squeeze itself in and escape to the
Yeah, the infection can escape too. Yeah, that could happen. Yeah. I mean, that's pretty good. You made it through this gauntlet, and you made it through that gauntlet, and you made it out. That's pretty good through the infection. Blood-borne infection. All right, thymus. This is where it's located. It's very large on a young child, say a 2-year-old, and it kind of atrophies as an adult. It's right over the heart pericardium area. The T and T cell comes from thymus. This is for T cell. There's a process called, we call it T cell education. It happens here. It's more of a 2-step selection process to select the naive immunocompetent T cells to enter circulation. two-step selection process that allows naive competent T cells into circulation. Here's the histology of the thymus. It's not in your study list to study, but I have a picture of it here. The idea here is that you have the cortex and the medulla. The cortex, the T cell education that occurs in the cortex is that you have reticular endothelial cells that are producing hormones like thymosin, thymopoietin. They facilitate immunocompetence. So these cells, they surround blood vessels and they form a blood thymus barrier. And that protects T cells from premature activation because the T cells that are here aren't ready. Okay, you shouldn't activate them. They may cause trouble. So you want to protect them from the blood. So the important thing to note here is that the reticular epithelial cells form a blood thymus barrier in the cortex. Reticular epithelial cells form a blood thymus barrier to protect immature T cells from premature activation. The medulla is a little bit lighter. What I want you to know there is that you have the same endothelial cells, particular endothelial cells, but they basically don't form that barrier. And immunocompetent T cells, they can migrate from the medulla, from the cortex, and enter the circulation. Okay, so the ones that can work will enter the bloodstream in the medulla, not the cortex. So I'll say in the medulla, there is no barrier competent T cells can enter circulation. Basic idea there. So what T cells do you want to let in circulation? Again, you got to select the right ones, and the selection process is twofold. You have a round of positive selection followed by a round of negative selection. Only T cells that make it through both rounds of selection can make it into circulation. Let me kind of talk about that. This T cell education. I'll put air quotes around the education. I mean, it's not like the cell is gaining information. It's like, it's more a development of self-tolerance. If you're a competent T cell, you're a T cell that tolerates self. You don't attack yourself. You tolerate self. You only activate in response to non-self. Okay, so that's what I mean by development self-tolerance. And again, 2 rounds. It's a 2-round selection process. The first round is positive selection. Oops. Positive selection, ask yourself this thing: does the T cell react to MHC 1? Put it in the form of a question. It's pretty much a yes/no question. Okay, before I answer it, you can see the answer up there. Which cells display class 1? Yeah, CD8s interact with them, but what cells display them? Remember, there's the APC, but this one is actually all cells. Remember when I said that? All cells display the class 1, except cells that don't have a nuke, like red blood cells. Okay, so think about it. You're trying to develop self-tolerance. Let's see if you can tolerate the MHC that like all cells have. Right? That's a good selection process. So, okay. If the answer is yes, the cell lives. You can you recognize self. You can interact with this molecule. The no's, if you don't, you're not gonna, you can't do anything. Okay? Apoptosis. Second P is silent. Apoptosis. Second P is silent. All right, the second selection process is the negative selection. The question I have there is, does the T cell react to a self antigen? Self-AG. So we got all the ones so far that can recognize MHC1. That's good. But now, do you want them to react to yourself? No, you don't. That would be an autoimmune response. So, if the answer is yes, those cells undergo apoptosis. If it's no, that cell lives. So what you've done is you've selected all the cells that can recognize your MHC1 that don't react to yourself. So that's what we call the naive immunocompetent. And yeah. Okay. Here's a picture of that selection process of the round 1, round 2. Here's round 1. Positive selection must recognize MHC. Okay. If it can't bind at all, well, you're no good. You can't even recognize the histocompatibility complex. Just no, don't need you. If you can recognize it, you can at least loosely bind and recognize it. Okay, yeah, you can live on to the second round. So in the second round, everyone can recognize MHC1. However, if you recognize MHC1 but bind too tightly to a self-antigen, that's going to kick off signal 1. Okay? You don't want that. You don't want to react to self. So that's a no, you get rid of it, apoptosis. So if you can recognize MHC1, you may be binding a little less tightly, put a little gap there. All right? So you don't kick off a signal. It's like, okay, hey, that's self, I don't react, I move along. That's what you want in your circulation. And those are allowed to live and they enter the circulation.
So that's what happens when you have immune response problem, like.
Yeah, that could be one of the
thyroid
things.
to
The T cell is reacting.
hypothyroidism.
Oh, I'm sorry, I cut you off. What did you say?
I was saying that does that cell bind too tightly when you have autoimmune response, like the hypothyroidism, which is called Hashimoto's disease?
That particular disease, I don't know. But the idea is yes.
Okay.
Yeah. Yeah. Or it could be for B cells. You have a clone of B cells that react to a virus, and you make a protein, an antibody, that kills the virus. But that protein is very similar. It also recognizes, say, cells of the beta cells of the pancreas that secrete insulin. And you attack yourself. Because the antibodies, all they do is they just bind to atoms that are similarly fit. That's an example of B cells causing the autoimmune response. Okay. All right, so I want to get you ready for that. We're going to take a break. Before we take a break, I want to show you the slides that I want you to look at in the lab. So, let me pull those up here.
Professor?
Yeah,
Can I ask you a question about the spleen?
go ahead. I'm listening.
How are people able to live without a spleen?
It's harder to find infection, but you have other lymph tissues, like all the lymphoid tissues and all the lymph nodes. I'd have to look up how they treat people who've had a splenectomy. But yeah, you can live. There's other lymph tissues that can fight infection for you. You can receive plenty of antibiotics or other things to help you out. Yeah. I should mention where the spleen is.